Hi class, welcome to the third lecture of the Medical Parasitology Lab. Today we're going to be talking about Trypanosomes and Leishmanias. So the first parasite we're going to be talking about is Trypanosoma brucei rodentiae. We're just going to refer to it as Trypanosoma brucei from now on. So it's the causative agent of the East African sleeping sickness, which is also known as African trypanosomiasis. This is an acute infection, and eventually it can be fatal if it's not treated. The vector for this disease is known as Glossinia, or the tsetse fly. A vector is just a living organism that can transmit infectious pathogens between humans or from animals to humans. And something that's important in identifying Trypanosoma species is the kinetoplast, which is just a circular network of DNA containing mitochondrial genes that we're going to see more later. So going into the life cycle of Trypanosoma brucei, so the way you first get infected with this parasite is a tsetse fly bites a human and takes a blood meal from the human. And then when that happens, metacyclic trypanomastogotes get injected into the human's bloodstream. Once they're inside of the bloodstream, the metacyclic trypanomastogotes then transform into bloodstream trypanomastogotes, and then they can then just be carried to other sites through the blood. They can also enter the lymphatic system through the bloodstream. They eventually reach the lymph nodes or the spinal fluid, and they replicate through binary fission while they're moving through the bloodstream and entering various sites. So during the acute phase of disease, which is just a shorter phase of the disease, trypanomastogotes can be found within the bloodstream. But once you enter the latent phase, which is when the parasite begins to enter other organs, they then become undetectable in the bloodstream. So the cycle then repeats itself or continues when another tsetse fly bites an infected human and takes a blood meal from that human. It then ingests the bloodstream trypanomastogotes, and once inside the midgut of the tsetse fly, the bloodstream trypanomastogotes become procyclic trypanomastogotes, and these procyclic trypanomastogotes also multiply by binary fission. Once the procyclic trypanomastogotes leave the midgut and begin to move closer to the salivary glands or the mouth of the tsetse fly, they'll then transform into epimastogotes, and then the epimastogotes multiply by binary fission in the salivary gland, and then once they're mature they become metacyclic trypanomastogotes, and then that is then the form that gets injected into the human when the tsetse fly then bites the human. Just a diagram showing a T. brucei trypanomastogote. So here they have a single flagellum, and then they have an undulating membrane with a single flagella attached to it that they use for movement. Their kinetoplast is at the posterior end, which you can see it appears dark purple in stainings, and then their nucleus is located pretty much in the center of the cell. And the trypanomastogote stage is the only stage that's found in humans, so you won't find the epimastogote inside of humans. They also have here a parabasal body, which is where the flagellum connected to the undulating membrane originates from. Here's a video just showing T. brucei's movement and how they swim. Here's just showing the process of their replication and how they divide their genetic material. So the kinetoplast DNA is always replicated before the nuclear DNA, or the nucleus DNA. So just this picture shows K is a kinetoplast, N is a nucleus, and it just shows here that the kinetoplast multiplies first, which is just overall something interesting and something that's pretty characteristic of Trypanosomes. So moving on to the T. brucei epimastogote, here we can see some of the key structures from before have moved. We still have an anterior flagellum, we still have an undulating membrane, but it's a lot smaller. The basal body has now moved to the center of the parasite instead of at the posterior end of the epimastogote. So here on the right side is the trypanomastogote that we already saw. We're just comparing them, so you can see very clearly the difference between the size of the undulating membrane, the location of the basal body, and also the location of the kinetoplast has also changed. It's also towards the center of the cell in the epimastogote instead of the posterior end in the trypanomastogote. So moving on to Trypanosoma cruzi, this is the parasite that Dr. Maldonado studies, so I'm sure she's going to tell you a lot about it in class. So T. cruzi is the causative agent of Chagas disease, which is American trypanosomiasis, which it actually is already starting to make its way into the United States. So the vector of this parasite is known as a triatamine, or the kissing bug. They've also been seen in El Paso. Something that really differentiates T. cruzi from T. brucei is that T. cruzi has an amastogote form, and that multiplies within the host tissue, and T. brucei doesn't have an amastogote form. So we'll see what that looks like right now. So here's the Trypanosoma cruzi life cycle. So similarly to T. brucei, it starts out when the vector, in this case the triatamine, takes a blood meal from a human. What it does is, instead of the parasites actually being passed by the bite itself, the parasite is actually passed through the feces. And so what happens is when you get bit, the triatamine defecates, and so what happens is that bite wound can get irritated and just feel itchy. And so what happens is if you scratch the wound, the parasite and the feces then gets mixed into the wound and actually enters the wound. And then so that's how the parasite actually enters the body. Once the metacyclic trypomastogotes have entered the body through the wound site, they then are going to enter tissue cells. And then once they enter those tissue cells, they transform into amastogotes. Here they are. They're these very small organisms. And so the amastogotes multiply by binary fission within tissue cells. And so eventually what happens is the intracellular amastogotes, or the amastogotes within cells, they do eventually transform back into trypomastogotes once they mature enough. And then when enough of them have multiplied, they then actually burst out of the cell and will destroy the cell that they're inside of. And this cycle continues. The trypomastogotes then, once they burst out of a cell, they then move into another cell. They transform back into their amastogote stage again and continue to multiply. And then eventually back into the trypomastogote form again, and then they burst out of the cell. And this causes just a lot of tissue damage in this process. And then so a little triatamine, or a kissing bug, comes along again and then will bite a human that's infected, and then that's how the triatamine gets infected with the parasite again. So then trypomastogotes then transform into the epimastogotes once they're inside the midgut of the triatamine. The epimastogote form is then what survives within the midgut of the triatamine. Then these epimastogotes multiply within the midgut of the triatamine, and then they transform into the metacyclic trypomastogotes, and that's when they move over into the hindgut, and then they're then ready to be excreted in the feces of the triatamine. So a key distinguishing feature of the T. cruzi trypomastogote is they tend to roll into a little C shape. You can see here they're kind of rounded in shape. Also this one too. The kinetoplast end is kind of rolled into a C shape. So that's something that really helps differentiate them from T. brucei. And then a characteristic that is shared between both T. brucei and T. cruzi is both of them can be found in the cerebrospinal fluid. And then similarly to T. brucei, they also have an undulating membrane. They also have the kinetoplast at their posterior end. So then moving on to the T. cruzi amastogote, here is a tissue staining of them, and you can see quite a few of them within the tissue. So they're only found within mammalian tissue. You're not going to find them within the triatamine. And when looking at them through a light microscope image like this one, you're only going to see the kinetoplast and the nucleus visible. So the nucleus would be this larger purple dot. The kinetoplast would be the smaller purple dot. And so here's showing the T. cruzi epimastogote. This is the form that divides within the triatamine midgut. So this form is only found within the vector. This form is not found within the mammal host. So they also have an undulating membrane that attaches closer to the center of the body rather than the posterior end. You can see that the nucleus is a larger purple object, and then the kinetoplast is a smaller purple object. And so here's a better visual comparison between T. brucei and T. cruzi trypomastogotes. So you can see that the size of the kinetoplast is really different between the two of them. T. cruzi's is a lot larger, and then their location was also a little bit different because T. cruzi's is a lot closer to the posterior end, or the back end, where Trypanosoma brucei has a little bit of space between the absolute posterior and then the actual kinetoplast. So overall a kinetoplast, and then that little distinctive C shape is a good way to differentiate T. brucei from T. krusei. So this video is just showing the T. brucei trypomastigote undergoing binary fission or division. So T. brucei trypomastigotes will be seen dividing within the blood of the mammalian hosts, but you'll never see trypomastigotes of T. krusei dividing in the blood of mammalian hosts. So moving on to the last parasite of today, it's Leishmania. So Leishmania is the causative agent of Leishmaniasis. The vector is known as the sandfly. So the most common form of this disease would be cutaneous Leishmaniasis, which just causes skin sores and skin lesions. There's other forms of the disease known as visceral Leishmaniasis, which is when the parasite infects internal organs. They usually go to the spleen, the liver, and the bone marrow, and when this occurs, it can be very life-threatening. There's also mucosal Leishmaniasis, which is the least common form, and this is when Leishmania parasites migrate or spread into mucous membranes. So here's the life cycle of Leishmania. So here on the left side you have the sandfly stage. So the way humans get infected is the sandfly takes a blood meal and bites a human. They inject the promastigote form of the parasite into the human skin, and so what happens is the immune cells, known as macrophages, actually will react to these parasites, and the way they defend the body is they phagocytize, or they kind of eat foreign objects, and so these macrophages will then phagocytize the promastigotes, but in a way the promastigotes actually use this to their advantage, because once they're inside of the macrophage, instead of being destroyed, the promastigotes instead are able to transform into amastigotes. And then the amastigotes then divide and multiply within the macrophage, and then eventually they can burst out and then spread into other tissue types and different cell types, and then invade those different cell types, and then multiply and then burst out of those different cell types, that's how they then cause the visceral form or the mucosal form of the disease. And so the way the sandfly will then get reinfected, or a new sandfly will get infected, is the sandfly then takes a blood meal of an infected host, and then they ingest the macrophages that are infected with amastigotes, which you can see here in number 6. So then once the sandfly has ingested these infected macrophages or other cell type, the amastigotes will then transform into the promastigote within the gut of the sandfly. And then the promastigote then divides within the gut of the sandfly, and then they migrate to the proboscis, which is that pointy mouth that flies have that they use to pierce your skin with. So they migrate to the mouth of the fly, and then the cycle just repeats itself. So here's some images of the Leishmania promastigote. So they have a singular flagellum, but they lack an undulating membrane, so that's a good way to differentiate them from Trypanosoma trypomastigotes. And then something that's also unique to Leishmania is the kinetoplast is located at the anterior end, or the front of the promastigote, the side that has the flagellum, where Trypanosoma had the kinetoplast at the posterior end, so that's also a good way to differentiate them. And so here's the Leishmania amastigote, which you'll find them within macrophages of the host. They have a large nucleus, a very small kinetoplast, and they have very short, small flagellum that are coiled up within the cell. You can't really see the flagellum within light microscopy. You can only see them through a more detailed microscopy like electron microscopy here. And so here in this image, it's kind of hard to see the kinetoplast because they're so small, but you can really make out the darker, larger purple dot would be the nucleus, the smaller one would be the kinetoplast, and then this huge, very large purple section here is actually the nucleus of the macrophage. It's not part of the amastigotes. So overall, here's a visual summary of the different morphological stages of Trypanosomes and Leishmania that we covered today. So you have the amastigote, which is only found in Leishmania. So our large oval represents the nucleus, our small oval represents the basal body, which is where the flagellum attaches to. So here we have a large nucleus, we have a large basal body. There's also such thing as Leishmania paramastigotes, but they're extremely rare and they've only been described in the literature briefly, and their function is unknown, so we're not really going to focus on them because there actually isn't really any pictures of them in clinical specimens anyway. So then we have the Leishmania promastigote. It just has a single flagellum attached to the basal body with the nucleus at the center of the cell. There's no undulating membrane. So here we have the epimastigote, which is found in Trypanosoma brucei and Trypanosoma cruzi. So they have their nucleus and they have their basal body closer to the center of the cell, and then they have their single undulating membrane. And then here is the trypomastigote form of T. cruzi and T. brucei. So they have their basal body closer to the posterior end of the cell, and then from there the undulating membrane spreads out and exits at the anterior end of the parasite. And that's the end of today's lecture. I hope you learned something new, and I hope you have a good day.